Okay, about 50 of you have joined. So we'll just continue epithelium and talk about glands. I'll keep it short. I have things to do for your senior batch, and then we will come back to the next tissue, which is connective tissue, in the next class. But we will continue with upper limb classes from tomorrow. So I'm just doing what we took on Wednesday, I think, was a histology, general histology. I'm continuing with that today. So what we had said was that the cell layers and shapes are what determine the epithelium in terms of how many cells, what is the density of cells, and what is the layout of the cells together. So we know because of the cell layers, cells placed in layers, where the bottom layer or the basal layer is always on a basement membrane, which is a connective tissue domain, and there may be more than one layer stratified, layered on top of the basal layer. This flattening out, even if the cells are tall or square or cuboid in nature, this flattening out of the entire tissue as such allows for diffusion across the sheets. So that's one of the functional characteristics, which is why this particular shape has been taken up by the cells. Similarly, it allows for filtration. For secretion, you need a more chubby cell so that there are no more cytoplasmic organelles. There should be space for the organelles to do their thing. So you need endoplasmic reticulum for the protein components. You need vacuoles. You need Golgi apparatus to secrete and put together and formulate the larger molecules which will be secreted as a product of the cell, which will be utilized either in its immediate environment or it will be delivered somewhere via a duct, and it will go and do its job somewhere else. To be able to do a secretion, you need some certain cytoplasmic space for the organelles to be there and to do their job. So at that time, the shape of flat cell will not work. So most of the secretory cells tend to be columnar in individual shape. But the tissue as such will still be in a sheet. So it will be a sheet of columnar cells. In terms of geometry, that is a most efficient shape for a secretory surface to empty its secretions into wherever the action, the enzymatic action or whatever is going to take place. For absorption, again, a thinnest possible layer is useful so that there is a minimum barrier for movement of molecules across a domain. Protection again requires a somewhat thickish sheet, but also requires characteristics such as keratinization or very, very tight intercellular junctions so that it doesn't permit any protein or molecule or living thing, virus, bacterial, fungal movements across. The cell surfaces, when they have to do absorption, are increased into microvilli. When they have to do movement of a sheet of secretion such as mucin, they are modified into cilia. Cell connections, I said, have to be very tight, very specialized cell junctions, which are not seen in other kinds of tissues so much. And today we will talk about glands. Epithelium as such, when it is part of another organ such as skin, which is doing mainly protection, will modify itself as per the function of that organ, right? And very often the function required is secretion, and then the modification of epithelium, or rather the transfiguration, the configuration taken up by epithelium becomes what we call glands. if this epithelial tissue, which is of a glandular nature, also has ductular nature, then we call them exocrine glands. And if it is developed without any ducts but still does its secretion and delivers its secretion to its target organ through some other method, which is basically through the venous blood, venous drainage, then it's called an endocrine gland. So we looked at cell connections in the last class. I won't go into that today. And this is particularly important when it comes to the function of protection, right? But it is also very important because when this very tightly interconnected sheet is present, it allows the cells to become as thinly laid out as possible also. So it also subserves the functions of absorption and filtration, which is an epithelial function. So let's come to the exocrine gland. So by definition, these are modification or configurations of epithelium which have happened for the purpose of creating products, cellular products, which will be used outside the cell. They are not necessary for the cellular metabolism. They are necessary for doing some job outside of the cell. This could be by just a single gland or by a collection or a single cell or by a collection of cells. So when it is unicellular, like we said, we saw in pseudostratified ciliated columnar epithelium, which we said lines the respiratory tract, right? We said that most of the cells are simple columnar ciliated, but they look pseudostratified. They look stratified because some of them are short and some of them are tall. And the short ones are also on the basement membrane and the tall ones are also on the basement membrane. But all of them are doing the same function, which is essentially going to become a ciliated cell which will move the mucin on the surface. But where will this mucin come from? So interspersed between these ciliated columnar cells will be singular cells called goblet cells, who will not have cilia, whose function is to secrete this mucin. They are protein producing cells, and singularly they keep on pushing this mucin out, extruding it out, which will go and sit on the surface, on the luminal surface. And it will just sit there unless the cilia move it. So concerted, coordinated ciliary reaction from the luminal surface of all the remaining columnar cells, the majority of the cells, will move that sheet of mucin in a particular direction. In the respiratory tract, it is away from the lungs, up the tree and out into the oropharynx and the nasopharynx, which will either be spat out or coughed out or run out of your nose as a whole bunch of mucinous sticky secretion which has already trapped any particulate matter which has gone in through your inspired air. We'll do this in more detail in respiratory physiology and when we do dissections of the thorax and the head and neck for exposing the parts of the respiratory system. We touched upon it in systemic anatomy when we did general anatomy of the respiratory system. The single-celled gland, which is shown as an example, is this one, the goblet cell from the respiratory tract. This is also present in the intestinal tract, right? Large and small intestine. Now, in large and small intestine, largely we have columnar cells, but they're in the ileum and in the jejunum, their majority function is absorption. So the simple columnar epithelial sheet is going to have a lot of microvilli on the surface, which are going to absorb whatever is being digested from your food. But apart from that, in certain areas, especially when the food is getting more and more absorbed and getting less and less liquid, getting more and more solid because it's getting dehydrated, because water is also getting absorbed. By the time it arrives in the large intestine, colon, appendix onwards, appendix, cecum, ascending colon, transverse colon, descending colon, rectum, it is becoming feces. It is getting more and more dehydrated, and in large intestine there is no further absorption except of water and electrolytes. So the material in the lumen is becoming more desiccated, and to move it, the peristalsis, which is going to go as a muscular wave on the smooth muscle of the intestine, is helped by also lubricating the semi-solid mass by providing a sheet of mucin on the inside lining of the gut. So who will produce of mucin? Again, you will have unicellular goblet cells interspersed in between the simple columnar epithelium of the large intestine, which will secrete mucin, which will lie on the luminal surface of the entire, especially large intestine, and allow a smooth passage of the contents, which are slowly becoming the feces of that individual. So for a long distance, many inches, many feet, this luminal content will be pushed by peristaltic wave, and it will be lubricated by the mucin secretion of the goblet cells, which are unicellular glands which are lining the gut. I'm giving you this much of background just to set the context. What you have to remember is the glands, which are epithelial structures. Unicellular, called goblet cells. Multicellular exocrine glands. These are a collection of cells of epithelial origin, which together do the same function, produce usually the same molecules. We classify them as per their structure, as per the structure of the collection of cells, not so much by, you could also classify them by the product. You could say protein secreting glands, or you could say steroid molecule secreting glands, or you could say hormone secreting glands, or you could say catecholamine secreting glands, whatever. But that is one way of doing it. There is no better way or worse way, but we found that structural classification helps, particularly when we're looking at disease processes next year. So this is stuck, this is international. Another way of dividing or classifying glands is their method of secretion. So some of them, the cell, like the goblet cell, will create a molecule of mucin or a collection of mucin molecules, like a bubble inside the cytoplasm, and then extrude it through the luminal surface. Right? So it forms a secretory vacuole, and then the secretory vacuole is extruded out of the surface, and the cytoplasmic membrane reforms over that after that vesicle is shed. Sometimes the vesicle plus a little rim of cytoplasm is lost from the surface, which means a little bit of cytoplasm is also lost along with the secretory vacuole. Sometimes the whole cell disintegrates, emptying its contents. The cell has to die, right? Because the nucleus will also get, everything is opened out. So there are different ways of secretion of any particular cell. So that is one way of classifying exocrine glands. We will come to that. You must have heard of ecrine. I don't know if you've heard these words, but we'll come to that later. But right now, just let us look at the structural classification of exocrine glands. So if the gland is a collection of cells which are going to, this is the surface. Usually we said, no, all epithelium has to be in sheets, and there's going to be a surface. So there's going to be a basal side of the cell, and there's going to be a luminal or surface side of the cell. So when that whole surface side of the cell is dipping under the level of the sheet and coming back, it is forming like a finger-like pocket, like a hole in, suppose you have a ground, right? Like one hole in the ground. So the earth, the surface earth, is dipping in and dipping out again. So when you see it in section, this is what it will look like. It will look like a U, right? So actually it is a finger-shaped, or like the finger of a glove. It's a dip into the surface, and it will be surrounded by the connective tissue of that organ, right? So the simple straight tubular glands are seen. One example is in the stomach, another example is in the colon. So the straight tubular glands usually are in a single sheet, which is why we are calling them simple. We always said if there is simple epithelium, it means it is in a single sheet. So simple straight tubular glands. If that aperture from the surface is singular, but the tubular pockets are more than one, right? We call it simple branched tubular glands. So you have one or two or three finger-like pockets, but you have only one mouth from which all of them empty onto the luminal surface. So that's called a branched tubular glands. The upper part of the stomach has simple straight, whereas the lower part of the stomach has branched tubular glands. What is secreted by them? It depends on the organ, and we will study that in detail when we come to systemic histology. Right now, just get the classification. Sometimes this tube-like extension from the surface becomes so deep and that in the surrounding connective tissue there is no further place for it to go straight. So in growing further, it curls upon itself and makes a coiled structure. That is called a simple coiled tubular gland. And it is also seen in stomach and small intestine. So when you see the gland cut section, if you see this cut section, it looks like a simple tubular. But if you see the bottom section, it may be cut here or it may be cut here. So it looks like round cut sections. We can't say whether it all belongs to the same tubular coil structure or different glandular cut sections all adjacent to each other. We can't say which sections belong to which of the coiled tubes, but sometimes they are spaced far apart. So if you see one mouth or one duct of the gland and you see several cut sections here, we can assume that all these cut sections are because of the coiled nature of the bottom, deepest part of the gland. And the singular longitudinal section you are seeing is because of the neck of the gland, which is emptying to the outside. If the bottom part of the pocket is not straight looking or tube like, but is looking more like a bubble or a balloon, right? The word we use for that is acinar or alveolar. Yeah, that can get confusing, but just remember both the words. Acinus and alveolus means a rounded, bulgy, ovoid, spherical, spheroid shape of the tip of that tube. So the whole tube is not of a same dimension. The bottom of the tube is much wider. It's like a sac or like a chamber or a space. So that's called an acinus or an alveolus. And the simple acinar, where you have a single opening and a single bottom, but that bottom is a bulgier space, and as you can see, the lining cells are also bigger in size compared to these cells, isn't it? And the lining cells, because of the shape of this acinus, they become wedge shaped. The base of the cell, here can you see, the base is wider, the apex of the cell is pointed. So if you look at each one three-dimensional cell is like a wedge. The bottom is wide and it has an apex towards the lumen. And all these wedge-shaped cells together form this acinus shape of that gland. But it is a simple because it is only a single sheet of cells. Simple acinar gland. The classic example of that is the oil-secreting glands in your skin, sebaceous glands. Then the same acinar thing could be a branched structure. Just like this one was branched, the outlet is the mouth. And it has got 2 or 3 feeding tubes. Here it has got 2 or 3 feeding acini or alveoli. Right? So you have 4 or 5 balloons doing the secreting, emptying into straight narrow parts which all together join and exit out of one opening. So that's why it's called branched. So simple branched acina. Again, sebaceous glands can be of this type too. In certain parts of the skin, they're more prolific. You know where your oily areas of your skin are. There you tend to get branched acinar glands. Compound tubular glands are the opening to the surface where the product is going to be emptied is singular. It leads into 2 or 3 ducts, each of which leads into 2 or 3 ducts. So there are 2 generations at least of ducts before you reach the actual secretory part, the bottom, the secretory parts, which may be of a simple tubular. These are the tubular nature, right? But we don't call it simple because they are not emptying straight onto the surface. They are emptying into one tube, which is emptying into another. And that is finally opening on the surface. So this part makes it branched. But we don't use the word simple anymore. We say compound because the tubular system itself is branching in at least 2 generations. If there was only a single branching, we call it simple branched. If there is more than a single branching, we call it compound tubular, such as the mucous glands of the duodenum. If the secretory unit is an alveolus or an acinus, that is a dilated part with wedge-shaped cells, and each of them is emptying into a common space. 2 or 3 of them are joining up and then emptying onto the surface. So it becomes again a compound branching because there are more than one level of branching. And the secretory units are alveolar. So it is a compound acinar. Classic example is mammary gland, which secretes milk. Then you have the most common type of gland, actually, pancreas, salivary glands. All the big visible glands in your body, most of them are of exocrine nature, are of this type, where the secretory unit could be acinar or tubular. In this, they're only showing acinar, but some units are tubular and some units are acinar. This is a common variety where the shape of the secretory unit is variable. Usually at the level of branching, all of them will be of one type. Then here, all of them will be tubular. Then here, all of them will be acinar, something like that. So here again, the illustration is not very accurate. But remember that tubulo-acinar means that the secretory units are mixed. So one unit could be tubular, the other unit could be acinar in shape. Right? But they are a singular gland. They will usually, the product is the same. The shape of the cell which is secreting that product may be different. That doesn't matter. The shape of the gland is called tubulo-acinar if the secretory units are a combination of tubules, long, slender, or acini, which is bulgy, ovoid. If they are joining up and then joining up again, that means there is more than one generation of branching, then we use the word compound. Okay. The classic example is pancreas or parotid, any salivary gland, any of the big ones that you know. So this is how we classify exocrine glands as per their shape. As per the secretion, which is what I was telling you before, how does each cell do its secretion is one way of dividing the gland. So there is merocrine, which means that you have each cell, each of these is a lining cell of this particular gland, and it is secreting through secretory vesicles, and these vesicles are going to empty the secretions into the lumen, and this is going to get into the duct, and this is all going to get out. So the vesicle releases the contents into the duct. Can you see the vesicle goes to the surface of the cell, and then the vesicle reaches the cytoplasmic membrane and then is emptied into the lumen. So the secretory products rise to the luminal surface in their vesicles and then are shed from the luminal surface. This method is called merocrine secretion, and example is sweat glands. Apocrine. The secretory products go to the apex of the cell, and the entire apex of the cell is shed. Like I said, so a little bit of cytoplasmic membrane, little bit of cytoplasm, maybe some cytoplasmic organelles, definitely the secretory vesicles are all shed into the lumen. Right? Can you see? So the tip of the cell is breaking off. It's like breaking off the tip and letting it go, and then reforming the cell surface cytoplasmic membrane. So the cytoplasmic membrane will have to heal, and it will have to reform itself because the tip of the cell is being lost. So here you can see there are no vesicles inside the cytoplasm. The product molecules were all floating around in the cytoplasm. They went to the apex of the cell, and the apex itself broke off and went off into the lumen. So in the lumen is all the apices of all the cells, which are all getting shed off, pinched off portions of the cell. This is called apocrine secretion, and this is true of mammary glands, what we saw. So as you see, mammary gland as per the old classification is, what was it? Remember? Branched, compound. It was compound acinar. All the secretory units were acinar in shape, and it had 2 or 3 levels of branching. Whereas in terms of classification by type of secretion, mammary gland is an apocrine gland. Then a third way of doing the secretion, first was merocrine, second was apocrine. The third way is holocrine. The entire cell sheds and should be replaced. Once a whole cell dies, then there's nothing left which will do the secretion. So it will have to be replaced by mitosis of some basal cells. And that methodology is followed by your oil secreting glands. So a entire cell with its secretion falls into the lumen, and this dying cell releases its secretory products. The cell is shed into the duct. And a basal cell, this basal cell, which was till now not doing anything, is by mitosis going to replace the surface cell which is being lost. Holo means complete. Holos is complete. So it complete destruction of the cell to be able to empty the products. These are three ways in which secretion can happen, and exocrine glands can be classified like this. Okay, so this brings us to the end of glands, and I've given you some examples. It's a small thing, not much in terms of, there's not that much to understand. But this is the basis of a good large chunk of pathology next year. Because many glands are the ones which undergo tumor formation. So you will be required to remember this, that they're classified like this, that this is how their secretion takes place. You will be required to remember the correlation with biochemistry, how cell function takes place, which part of the cell does what thing, what can go wrong, why would a cell undergo a malignant transformation, a metaplasia, right? Not necessarily malignant, but at least a hypertrophy, an increase in number or an increase in size or a increase in function, which will come, the patient will come to you as a something is not happening right. There's too much of something happening or too little of something happening. And then you'll figure out from their biochemistry what is too much or too little. And from that you will guess, you will surmise that, oh, falana thing, falana gland, falana area, falana tissue is having some hyper or hypo problem. Then you will send for an investigation such as an ultrasound or an X-ray, which will confirm whether your suspicion is correct or not. Then you will send for a definitive investigation called a biopsy. You might want to get into that particular organ which you're suspecting and take a little piece of it and look at it under the microscope and see what the hell is happening over there. At that time, you'll be required to remember the background. What type of gland was it? What was it supposed to do? What is a normal picture? Health, in health, what will it look like? So we will show you lots and lots of glands from different places. We'll show you classic examples like we gave examples in this class. But also otherwise, you must realize that any area, any tissue which makes a product which goes somewhere else, which doesn't serve the individual cell, but serves the neighbors or the next organ or the next system or goes far away somewhere and does something. All of them are glands, and we have loads and loads of them. Almost all metabolism is because of glandular function. So it's important to remember. We chose to classify it as per structure because that is very facile. It helps us in terms of understanding and in terms of reporting and in terms of sharing information with other scientists. But for the body, for the individual organism, all of them serve the master, which is the body. And if one or the other of them is hyper or hypofunctioning, there must be a place where dysfunction of control loops is happening. So you then have to correlate with the whole individual patient rather than that one little tumor in one little part of the body. Most disease is never an isolated thing. It's not, one little thing of your ear going wrong and the rest of the body need not know about it. It doesn't work like that. Especially when it comes to glandular function. There are lots of interrelationships, interdependabilities, which is why understanding metabolism and homeostasis takes time. It will take years for it to sit properly and to grow in your understanding. Right now, you need to know all the terminology. You need to know the hypothesis or the concepts of why we classify, what we call it, what we know about how it works. And slowly extrapolate it to, if this goes wrong, it can become disease, or this may go over or under because the body needs it. Then it is not disease. But at some point, that may go a little beyond the confining control mechanisms of that tissue or that organ, at which point we will call it a disease. It's tricky. It's also very fascinating and challenging. I hope you'll enjoy the study of medicine as you get into the subject deeper and deeper. Anatomy tends to be very practical. You name it, you have to know what it looks like, you have to draw it right, you have to recognize it on the slide, you have to set it in your memory. All the parameters which make a cell healthy, or which indicate to you that the cell and the tissue is healthy, have to become background. They have to become so deep. Then any difference in parameter will suddenly appear to you when you look at it. Otherwise it won't. Your eyes won't even see what your brain doesn't know. Right? So entire first year we're just hammering normal into you. So that from next year abnormal will appear because, oh, this isn't what I saw last year. Right? So how is it different? It'll take you time to learn that. And then you'll figure out, oh, there are 18 ways of being different. And then the whole arena of disease will become real. Okay, but always, however big that arena of disease is, the baseline of health is actually larger. And the beauty of health is something you should never lose sight of. Okay, I'll stop right now. It's been a short class, but we usually keep glands as a separate class and don't mix it up with anything else. Look at this classification. It's a very sweet little classification, isn't it? Easy to see. I hope you've got this. Is there in every histology textbook, any atlas, anything you might have purchased, or if you go online, you'll see it. You can also browse a few slide pictures. These are paintings, actual pictures. They won't look like this. The actual tissues will not look like this, right? These are conceptual diagrams, because when you actually see the slide, it's all together. There is epithelial tissue, there's connective tissue, all sorts of things, and it's a very low magnification. And it won't be with these colors either. There'll be more pink and purple colors. You'll have to learn to see that this is the basis of what you're looking at. Okay, so learn this right, learn the terminology. Get the classification very easy, it's a no-brainer actually. But recognizing it in a real tissue is a little bit of a translation challenge, which will happen when you come for practicals. At that time, we will revise this again and show you the real thing, and then you'll learn to correlate this and that, draw the diagram, do the labeling. Then somewhere at some point that penny will drop, oh acha, like that. Okay. So I hope we'll get you on campus soon so that we can show you the practicals. So I'll stop now. Tomorrow there'll be a upper limb class, and then we will continue. I would also like to start general embryology. One of our staff members will probably take it up. I'll put it up as a timetable. I was actually waiting, hoping the college will announce your arrival on campus. If that's not going to happen, then the rest of this month will be only online classes. In which case, we will find some way of introducing you, at least through videos, to dissection. I'll attempt that in my next class. Thank you for now, and come tomorrow for tomorrow's classes. I think today's is done. Bye. Have a nice Monday.